morning, girls. Today let us study about oogenesis. Oogenesis is the formation of ovum or egg from the undifferentiated germinal epithelium lining the ovary. Oogenesis begins in the fetus prior to birth, that is, when the fetus is developing in the womb of the mother. Oogenesis is completed in the following three phases: multiplication phase, growth phase, and maturation phase. Multiplication phase begins in the fetus prior to the birth. In multiplication phase, the primordial germ cells undergo mitotic division to produce large number of diploid oogonial cells or egg mother cells. The oogonial cells undergo mitotic division and projects as tube-like structure or cord-like structure called egg tube of Fluger. The egg tube of Fluger then arrange themselves as mass of cells and is known as egg nest. Among these cells, only one cell will become the primary oocyte and the future egg. The rest of these cells are known as follicle cells. They surround the developing primary oocyte and provide nourishment. They are known as primordial follicle. The primary oocyte continues to grow in size. The growth phase of the primary oocyte is very long, extending over many years. At puberty, the cytoplasm volume increases and nucleus enlarges in size. Then, the primary oocyte undergoes first meiotic division to form two unequal cells. One cell is almost the same size of the primary oocyte and is known as secondary oocyte. The other cell is very small, having a very little amount of cytoplasm and is known as polar body. The secondary oocyte and the polar body undergo meiotic II division. The secondary oocyte gets arrested, or you can say that the meiotic II division get arrested at the metaphase II stage. The secondary oocyte is now released from the ovary, which enters the fallopian tube and awaits in the ampulla for fertilization. After fertilization, the meiotic II division is completed, thereby forming a large haploid oocyte. Then, inside the fertilized egg, the fusion of sperm pronucleus and egg pronucleus take place to form the diploid zygote. The three polar bodies which are formed gradually disintegrate. Here, this is the diagram of ovum at the time of fertilization. Here, we can see that it is at the metaphase II stage is being arrested. Here, we can see this is the secondary oocyte. It is surrounded by a plasma membrane which is known as oolemma. The cytoplasm which fills the oocyte is known as ooplasm. At the periphery or underneath the plasma oolemma, we can see granules are present. They are known as cortical granules. The oocyte or the egg remain surrounded by a protective covering that is known as zona pellucida. The zona pellucida layer is secreted by the oocyte itself. When the secondary oocyte is released from the ovary, some of the follicle cells remain attached to the oocyte to form the second protective membrane and is known as corona radiata. In between the oolemma and the zona pellucida, a small space is present which is known as perivitelline space. So this is the structure of the ovum at the time of fertilization. Thank you.